Again, myself, Dr. DEBANSU SEKHAR PARMANIK, reader in Zoology from DEVI College, BHUBANESWAR, and I am welcoming to you all to this YouTube class. My dear student, in my last class, I have told you and we have also studied regarding immunity. So, basic concept of immunity. In that class, we have defined what is immunity, how our body can fight against different type of virus, protozoa and antigens like structures. So, that we have already studied. So, in that section we have also studied that this immunity of our body is developed either through innate immunity system or innate immunity method, or it is developed through adaptive method. Again, during innate immunity method we have studied how different type of barriers are there. They are actually defending us, fighting with different type of viral particles, different type of foreign particles to protect our body. Similarly, we have also studied regarding the acquired immunity, and in this acquired immunity section we have studied how our T cells and bone marrow cells are fighting for our body. So, that we have discussed. So, today in this class we shall discuss about to some extent regarding antigen, what is antigen, what is antibody, what is immunoglobulin, then we shall discuss about vaccine, we shall discuss about AIDS, and we shall also discuss to some extent about allowances like that, which is actually assigned or which is given in this unit. So, first of all I am again briefing you regarding antigen. So, in our last class we have also encountered one word, that is antigen. So, what is antigen? Antigen is a foreign substance or a foreign particle which actually induce or which actually produce or when enters to our body, which induces the production of antibodies in our body. So, only one line definition: antigen is a particle, antigen is a structure, antigen is a foreign particle. When it enters to our body, it induces the production of our antibody. So, we shall again discuss about that in detail. So, this antigen may be a virus particle, this antigen may be a bacteria, this antigen may be a protozoa, this antigen may be a blood and red blood cells from other individual which has given to me. This antigen may be a snake venom. Whenever one snake bites to us, at that time the snake venom is introduced to our body. That is also one type of antigen. Allergens, different type of pollen grains like that, they are also allergens. And certain proteins present in food which is actually giving some allergy to us, they are also antigens. So, what I want to say, antigen means the particle which actually causes harm to us and that actually induces the production of the antibody in our body, that is an antigen. And chemically all these antigens are protein. Chemically all the antigens are protein, or chemically it is a protein, or it may be polysaccharide, it may be a nucleic acid. That means mostly it is one organic component, it is one organic molecule. And this antigen actually carries, that means if we study the chemistry of that one, it is again observed that this antigen carriage or these antigens have a number of antigenic determinants. That means on the surface of the antigen, if this is a protein particle, on the surface of this protein particle there are present some determinants which are actually called as epitopes, which are homologous to paratopes present in our antibody. That means what I want to say, in my body antibody is there. Whenever antigen introduced into my body, the antigens will be produced, and this antigen possesses a number of configuration, a number of structure upon its surface. These structures are called as epitopes, and these epitopes generally binds to the antibody, that means particular site of the antibody, and that site of the antibody is called as paratope. That means the antibody has paratope, antigen has epitope. So, whenever one antigen comes in contact with antibody, at that time the epitope of the antigen and paratope of the antibody combined or they react. So, this is the thing about the antigen. And mostly again we have observed that some chemicals are there, they are not complete antigens. That means they are antigenic in their nature, but they cannot induce successfully all the antibodies. So, that they are called as hapten. So, hapten is a partial antigen, and it cannot alone introduce or it cannot alone induce the antibody, but whenever this antigen type, that is hapten, combines with some another protein, at that time it is able to produce the antibody. So antibody is 100%, antigen is 100%, that means molecule 100% able to produce the antibody, but hapten is one type of antibody, that is a partial antigen. So this is a one type of antigen which is called as partial antigen, which cannot trigger or which is not able to trigger the antibody in our body, but whenever it will combine with one protein, that means combination with one protein, at that time the hapten will become the antibody. So antigen. So this is all about the antigen. So now we shall see, this is the structure of an antigen. For example, in this picture I am giving one slide that is having one antigen. For example, this is your antigen. These are the sites, or these are the portions which are called as epitope of the antigen, and this is one antibody. This is present in my body. This is a foreign particle which is antigen. This antigen has particular sites, they are called as epitope. This is one antibody. This antibody has the specific sites, they are called as paratope. The most important thing is that a particular antigen has a particular type of paratope, that is homologous, so that in that region or in this region the epitope of the antigen and the paratope of the antibody will combine. So this is the antibody antigen reaction. So this is all about the antigen and antigen. So now we shall come to the antibody. Then what is antibody? So again one short question. Antibody is also called as immunoglobulin. Immunoglobulin. Immuno means this is concerned with the immune system, and globulin means is a protein. So antibody is again a protein which is produced by our immune system. So they are called as antibody, and this antibody is shortly, that is immunoglobulin is also one antibody. This is shortly written as Ig. This is a protective protein, and it is produced by the immune system in response to a foreign substance that is called antigen. That means in our previous slide I have told you that the antigen is a protein which can induce the antibody. Similarly, antibody is a protein which is my protein, is a body protein, which when comes in contact with a foreign protein, at that time it will be produced. So antibody is a substance, or antibody is a protein which is commonly called as immunoglobulin, and it is produced by the immune system in the response of one foreign particle, or there is an antigen. So now how it is produced? In our previous classes we have discussed that our bone marrow cells, which are commonly called as B cells, and these B cells when come in contact with one antigen, at that time this become a plasma cell, and this plasma cell mature, and this plasma cell is nothing but one B lymphocyte. This plasma cell then mature. When the plasma cell is mature, at that time this plasma cell produces the protein. That means in our body the bone marrow cells are there. This bone marrow cells produces B lymphocytes, and these B lymphocytes when come in contact with an antigen, at that time they become matured. The mature cell is called as plasma cell, and this plasma cell then produces a number of proteins which are called as immunoglobulin. Clear? Now in general, immunoglobulin is a protein which is consisted of 4 polypeptides. 4 polypeptides means 4 protein chain, polypeptide chain. Each polypeptide is again containing a number of amino acids. We know amino acids when combined with the, or amino acids when attached with peptide bond, at that time this form a polypeptide, and this polypeptide is a primary structure. Then polypeptide fold upon itself and produce a secondary protein or secondary structure, then tertiary structure, and finally it become a protein. So this immunoglobulin is also a protein which is consisted of 4 polypeptide chain, and out of that 4 polypeptide chain, 2 chains are small, and they are small chains which are called as light chains, and 2 are longer or large chains which are called as heavy chains. For example, this is one immunoglobulin, and this immunoglobulin is consisted of 4 chains, 1, 2, 3, 4. Out of these 4 chains, 2 are called as heavy chains and 2 are called as light chains. I shall show you in our later slide. So 2 are heavy chains and 2 are light chains, and all these chains are joined with each other by the disulfide bond. There are different regions on that polypeptide, and they are commonly called as constant region and the variable region. So now you see this is the structure. For example, this is the plasma cell. When the plasma cell come in contact with the antigen, at that time this plasma cell produces a number of protein. That proteins are called as immunoglobulin, and immunoglobulins are otherwise called as antibody. So this is the structure of one antibody or one immunoglobulin. This immunoglobulin is consisted of, you see, 4 polypeptides. Out of the 4 polypeptides, these two red mark are there called as light chain, and these 2 are called as heavy chain. So this is assume one Y-shaped structure. These 2 are called as heavy chain. That means this is a polypeptide chain which is longer, containing more amino acids. So this is called as heavy chain or heavy polypeptide. So this is 2 in number. Similarly, this is a light chain, and this light chain contains a small number of amino acids. So this is called as light chain. So this is the structure. Again, you see, if we analyze detail, then again you see this is the heavy chain and this is the light chain. This portion of this heavy and light chain contain a region which is called as variable region, and this region is called as constant region. That means this Y-shaped polypeptide structure again contain 2 region. One is called as variable region and one is called as constant region. That means both the light chain and heavy chain have a particular area that is regarded as variable region, and this region is called as constant region. So this is just simple diagram to know about the immunoglobulin. So this is a typical structure of a antibody. Again, different type of antibodies are there, different type of immunoglobulins are there, they differ in only structure. We shall see in later slide. So this is all about the antibody or immunoglobulin. Now we shall see how many type of immunoglobulins are there. It is observed that in our body, in our blood, different type of antibodies or different type of immunoglobulins are there. They are known as immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin E and immunoglobulin D. So 5 major classes or 5 type of immunoglobulins are generally produced in our body in response to antigen. So they are first immunoglobulin G, which is shortly written as IgG. So these are the most abundant class of immunoglobulin. That means their number is high. That means 80% of immunoglobulins present in our blood in response to antigen are generally immunoglobulin G. So what they do, it protects the body against bacteria and virus found in blood and intestine and can cross the placenta from the mother to fetus. That means this is the only type of immunoglobulin which can pass from one mother to its baby when the fetus is growing inside the uterus. That means it can cross the placenta. So this is the only antibody which can pass the placenta. That means it can be transferred from its mother to its growing baby which is present in the which is growing in the uterus. So second one, the second important and second largest one or second abundant one antibody or immunoglobulin is the immunoglobulin A. So this is the second most, that means 15%, and this is abundant class of immunoglobulin found in sweet tear colostrum, and that means it is present in our sweet and it is also present in our tear from the whenever the tear comes out from the eye, that also contain this immunoglobulin A. That means they are protecting the ear, they are protecting the body, and they are also present in colostrum, that means the first milk of the mother when the baby nourishes. So it provides localized protection against bacteria and virus. That means these are the antibodies, these are the immunoglobulins, they are present in different localities and different locations, and they are providing defense to that particular organs or that particular anatomical organs present in the region. So third one is the IgM. So this is the largest immunoglobulin found in lymph. It activates B cell. So this is the antibody which is largest in its structure, and it is found in our lymph. It activates B cell. What it does, whenever it is or whenever one antigen comes to our body, at that time it activates B cell. That means this antibody activates the B cell and help in complement activation, and this is the only what antibody which is produced earlier. That means when the baby is growing, when the fetus, that means when the young is born, at that time this is the first antibody which is produced. That means earliest to be synthesized in the fetus. So this is the first one which is produced in our body. Then the fourth one is immunoglobulin E. So this is another type of immunoglobulin, another type of antibody which is known as immunoglobulin E. This is involved in allergic and hypersensitive reaction. So whenever we come in contact with any type of allergen, whenever we contact with any type of material which is not concerned with our body, which is completely foreign to us, at that time they make the allergic reactions and hypersensitivity reactions in our body. That means they reject that material. Then last one is the immunoglobulin D. So immunoglobulin D, which is the fifth type of antibody, found on surface of the B cell. So this is the only antibody which is present over the B cell. So this is a surface antibody. That means it is not present in the blood plasma or not present in blood, rather it is found on the surface of the B cell as antigen receptors and activates for antigen recognition. That means this is present over the B cell. So they are called as antigen receptor. So whenever one antigen comes in contact with that B cell, that receptors can recognize this antigen and finally make the reaction. So that is the immunoglobulin D. So these 5 types of antibodies are there in our body which are produced in response to antigen. So then what they will do? That means in our first slide we have discussed about the antigen, its property, its chemistry. Then in the second slide we have also discussed about the antibody which are produced in our body in response to antigen. Then in this slide you shall see how these antibodies will react, how they will react with the antigen. So different type of reactions are there. Mostly they are agglutination, then opsonization, then precipitation, then neutralization, then lysis. That means these are the mode of action. So different type of actions are exhibited by different type of antibodies in our body. So first one is agglutination. That means this is the clumping of the microorganisms or the blood cells. That means whenever the antigens come in contact with the antibodies, at that time one type of reaction can be undertaken, that is called as agglutination, where clumping will be done. That means the cell will be clumped. So this is called as clumping. So clumping of the microorganisms, it may be bacteria, it may be virus, or sometimes the blood cell, at that time they will be clumped. So this is called as clumping. Second one is opsonization. That means whenever an antigen will come in contact with the body, or that means enter into the body, immediately antibodies will be secreted and the antibodies will coat the surface of the antigen. They will be cover the surface of the antigen. So when the antigen is entered, at that time antibody will secrete and that antibody protein will cover the surface of the antigen. That means when or once the antigen is covered by the antibody protein, at that time it is very much susceptible by the macrophage to be eaten. That means macrophage can react immediately with that type of cells. So this is called as opsonization. So opsonization is nothing but this is the antigen is coated with antibodies so that this opsonized antigen can be destroyed by our immune system very easily. So this is called opsonization. Third one is called as precipitation. So what I am discussing about how the antigen reacts with the antibody. So these are the different type of reactions. So third one is precipitation. Once the antigen comes in contact with the antibody, at that time antibody helps to cross-linked, so that the antigen will be cross-linked so that antigen cannot do harm. So this is called as precipitation. The fourth one is neutralization. That means the antibody will block the toxins of the viruses. That means if one virus is there invaded to our body and this virus is producing some toxic materials, then what they will do? They will neutralize, they will block the toxin secretion by that virus. So this is called as neutralization, neutralizing this bacteria, that virus. Then lysis. Sometimes antibody causes or antibody causing rupture of the plasma membrane of the bacteria or virus we have studied in our previous classes. So this is called as lysis, means destroy. So in this slide we have just briefly discussed about the antigen antibody reaction. So different reactions are agglutination, opsonization, precipitation, neutralization and finally lysis. So this is just a picture regarding this. So this is the picture of precipitation. That means these are the Y-shaped structures are the antibody, these red structures are the antigen. So whenever antigen comes in contact with the antibody, at that time they are precipitated. So this is precipitation, picture of precipitation. This is the picture of agglutination, and this is the picture of then neutralization. It is neutralized, and opsonization is there also. So opsonization means the antigen will cover the antibody, and finally this will happen, then opsonization will occur, and once it is opsonized, that means once the antigen is covered by the antibody, at that time this will be taken by the macrophage, and macrophage will digest this one. So this is one antigen, and this antigen is again opsonized by the antibody. So these are the antibody, and this is the antigen. So this is one macrophage. So when one antigen is opsonized by the antibodies, at that time the macrophage is easily, or macrophage can easily digest or destroy this one. So this is one cell lysis. That means when one antigen will enter into the body, at that time some antibodies are there, they will activate some proteins, and these proteins are called as complement proteins, and these complement proteins will make some pore upon the plasma membrane of that antigen, and finally that will be destroyed. So this is the lysis. So this is just a brief diagram how different type of reactions are going in connection with the antibody and antigen. Now we shall see about the vaccines. So this is all about the antigen and antibody and their reactions. So now vaccines. So you have heard one word, that is vaccine. So this is one structure, this is one chemical when it is introduced into our body to develop our immune system, we commonly know. So now in this 2 to 3 slide, we shall briefly describe what actually vaccine is. So vaccine is a preparation. So this is a preparation, this is a chemical, and this preparation is done from dead or weakened germs of a particular disease, which on injection to the healthy person provides active or passive immunity by inducing antibody formation. That means I am one individual having my immune system, but my immune system may not, or it is not completely working. So what can be done? Now a particular type of bacteria or a particular type of germ can be taken, and this germ is of dead type or died type, then it is killed one. So then this germ will be inoculated to me, or it will be injected to my body, so that what will happen, that germ, inactivated germ will produced, or this will induce my immune system. So vaccine is a preparation of dead or weakened germ, and it will be injected to the body. So when this dead or weakened germ are injected to the body, at that time this person's or the person will develop active or passive immunity by inducing antibody formation. That means vaccines indirectly induces the antibody formation. So this is called as vaccine, and the process is called vaccination. So vaccines generate, what they do? Vaccines generate memory B and T cells. Whenever they enter to our body, they actually generate the memory B cell and the T cells that recognize the pathogen quickly. That means if I am vaccinated now for a particular germ, and that germ when introduced to my body through the vaccine, it will produce the antibody, and that antibody are of memory B type cell or T cell. So whenever original antigen will enter, at that time they will fight immediately. They can recognize that antigen immediately because they have been seasoned previously by vaccine. So this is all about the vaccine. That means vaccine generate memory and memory B cell and memory T cell, which can recognize the pathogen very quickly, so that they can act very quickly. So this is all about the vaccine. Then we shall see types of vaccine. Different type of vaccines are there, and the first one, how the vaccines are prepared, the first one, or how they work in our body, from which source they are collected. So this is called as type of vaccination or type of vaccine. The first one is attenuated whole agent. That means this is called attenuated whole agent. That means the whole agent is given, use living but weakened microbes. That means for some diseases or from some bacteria or some viruses are there, they are collected, they are cultured, finally that means in the laboratory they are weakened, that means they are very weak. Then this weakened virus or bacteria are introduced to the body, so that they will produce the B and T cell. So this is called as attenuated whole antigen. that means, or attenuated whole agent, that means the whole agent will be introduced, that means whole organism will be introduced through the vaccine. But what will happen, this organism is weakened, this is not completely active organism, so inactivity, it has been inactivated. So the inactivated is called as weakened, and these microbes are once inactivated, they will be introduced to our body, which is called attenuated whole agent. And what type of vaccine as produced in this case? So they are sabin polio vaccine, that means the polio vaccine we are taking, they are from attenuated whole agent, that means the whole virus is again introduced to this one, but it is in weakened form. The second one is inactivated whole agent. So in this case the killed microbes are there, that means the microbe, a particular type of microorganism is taken, and this organism then will be cultured, and finally that organism is of dead type, that means the organism will be killed, and that killed organism will be introduced into the body as a vaccine. So the example is bacterial vaccine for pneumococcal pneumonia, then cholera and typhoid. So typhoid, cholera, these are different type of diseases, and these diseases are generally caused by different type of bacteria. So in that case what will happen, the bacteria will be collected, they will be inactivated or they will be killed, and this killed bacteria will be introduced in the body. So when they will be introduced to the body, at that time in the body they will introduce or they will produce the antibodies. So this is called as inactivated whole agent. Third one is toxide, that means in this case the vaccine is prepared by taking the toxins of the particular microbe, that means this microbe is able to produce a particular toxin, and that toxin is taken and that toxin is introduced as a vaccine. So the tetanus vaccine is the best example of this one. Sometimes this vaccine is conjugated with another type of chemical, so this is called as conjugated vaccine. So this is another type of vaccine, polysaccharides combined with protein. So a particular microbe is taken and this microbe contains some protein, and that protein is again mixed with the polysaccharide. So polysaccharide combined with the protein is producing one type of vaccine, that is called as conjugated vaccine. So these vaccines are generally produced, or these vaccines are generally manufactured for the Haemophilus influenza. So Haemophilus influenza vaccine are produced by this method. So last one is sub-unit vaccine, that means in this case neither the microbe is taken nor the killed microbe is taken, but a fragment of the microbe is taken. So use of the fragment of the microbe is taken, that means the whole microorganism is not taken, a fragment, a small piece, a broken piece of that microbe is taken and it is introduced into the body as a vaccine, and that will produce the antibody. So this is called as sub-unit vaccine, that means use of fragment of microbes. Example is vaccine against the hepatitis B. So different type of vaccines are available and for a different type of antigen. So this is the method how they are being prepared. So now we shall see in this slide the HIV and AIDS. So this is again very important one, because in this class we have discussed, or we are discussing about the immune system and immunology, and this is the virus, or this is the only virus which actually destroy our immune system. That means they make us very weak, so that the immunity power of an individual is been lost. So when immunity power of an individual lost, at that time different diseases come to the body. So hence this is called as acquired immune deficiency syndrome, or this is called as AIDS. So it is again we shall discuss in a very brief. So AIDS is again a disease. This is not a disease, this is called a syndrome, because a number of symptoms will be given, will be observed. So this is a syndrome. So the full form of this one is acquired immune deficiency syndrome. That means our immune system become destroyed, become deficient due to acquiring of some agents to our body. So this is acquired. So I am acquired, or I acquired some viruses, and that viruses are destroying my immune system, so that this is called as acquired immune deficiency syndrome. It is a disorder of cell mediated immune system of the body. So very important thing is that, so AIDS is a disease, or AIDS is a syndrome which is concerned with the immune system, and especially the cell mediated immune system, because in our last class we have studied the acquired immune system is again of two type. One is called as humoral immune system or humoral mediated, another one is called as the cell mediated. Humoral means B lymphocytes, they are producing the antibody which are circulating in the blood, so this is called as humoral. But the T cells which are producing different type of T cells, they are again protecting our body, so that is called as cell mediated immune system. So specially the AIDS is the destruction of the T cells of the body, not the B cells. So AIDS is also called as acquired immune deficiency syndrome, full form is acquired immune deficiency syndrome. It is a disorder of the cell mediated immune system of the body, that means T cell will be destroyed. Usually the T cells, what the T cells actually do in our body, the T cells generally stimulate antibody production by B cell. In our body, B cells are producing the antibodies which are, we have just discussed, they are the immunoglobulins. The immunoglobulins are produced by the B cells, but T cells support, help, or they activate the B cell to produce the what? The immunoglobulins. So the T cells usually stimulate the antibody production by B cells. Due to AIDS, the T cells are reduced, T cells are destroyed, hence individual loses the natural immunity. AIDS was first noticed in USA in 1981. So this is not one very disease of very long back or past. This is a recent disease. It was noticed in America, United States of America in 1981. South Africa is considered as world AIDS capital. The highest number of individuals are suffering in what? That is in South Africa. So South Africa is considered as world's AIDS capital. So a maximum number of people are there who are suffering from AIDS. AIDS virus was discovered by Professor Luc Montagnier in Institute of Pasteur in Paris in 1983. So the disease was discovered in 1981, but how the disease is caused, how the syndrome is caused, which was discovered by scientist Professor Luc Montagnier at Institute Pasteur in Paris in 1983, and he discovered that it is caused due to a virus. This is called as AIDS virus. Then AIDS infection was detected in India, especially in India, in 1986, especially in the state of Tamil Nadu and in Chennai among some prostitutes. So this disease was detected in India in 1986 among the prostitutes of Chennai in the Tamil Nadu state. So the disease came to the world in 1981. The virus was discovered by scientist Montagnier in 1983, and this disease was detected in India, especially India in the state of Tamil Nadu in 1986. So this is all about the AIDS. Now we shall see the virus. So how it is causing? So AIDS is caused by virus that is called HIV or human immune virus. HIV, human immunodeficiency virus. So this is HIV, it is a virus. So like other, it is a spherical virus, like other viruses, it is a spherical virus or it is spherical in shape, about 90 to 120 nanometer, very small particle, and its genetic, the most important thing is that we know whether it is a microbe or whether it is a bacteria or it is a cell or it is a virus, the genetic material must be the DNA. But this is a virus where the genetic material is RNA. So this is a RNA containing virus or RNA virus, not the DNA virus. So its genetic material is again two number of identical single stranded RNA, because RNA cannot be double stranded, DNA is only double stranded, but this virus contain two filamentous RNA, single stranded RNA, which are identical, and along with that the most important thing is that this also contain one enzyme. That enzyme is called as reverse transcriptase. So this is one enzyme which is also present along with these two RNA molecule. That means what I want to say, the HIV virus is a spherical virus of 90 to 120 nanometer in its size. It contains two identical single stranded RNA molecules as the genetic material. Along with the RNA, one enzyme, that means two enzymes are there, that means the enzymes are there present, they are called as reverse transcriptase enzyme. Clear? the virus has a lipid bilayer and projecting knobs of glycoprotein spike. So, if this is structure, spherical structure, then this is covered by lipid bilayer. This virus is covered by one lipid bilayer, and from the surface of that lipid bilayer, they are projecting some spike like structure. They are of glycoprotein in nature. Then beneath the lipid bilayer again present the protein coats. This is just the structure in brief. So, this is the structure. So, in this slide you see this is the structure where the glycoproteins are there. These two are the RNA molecules, and this is the reverse transcriptase enzyme. So, again this is the protein and these are the glycoprotein spikes. So, this is the clear picture, you see this is the clear picture again you see. So, this is one RNA, you see this is one RNA, this is another RNA, these two RNA are single stranded RNA. So, this is one molecule of reverse transcriptase, this is another molecule of reverse transcriptase enzyme. So, again this is the capsid or protein code, this is the double layer lipid code, and these are the what? These are the spike like structures which are called as or which are chemically glycoprotein and their spike. So, this is the structure. The most important thing is that the genetic material of this virus is two individual identical single stranded RNA. Along with those RNA there are present reverse transcriptase enzyme. That means this is one enzyme which can form DNA from the RNA. So, it is called as reverse transcriptase. Okay, this is all about the structure. Then transmission, how one individual will be suffered or how one individual will be caught this its disease. So, transmission, how it will transfer. So, this is transmitted or this is transfer or this is transformed from one individual to the other by infected blood. That means one individual is suffering from AIDS, in its blood this type of viruses are there. Whenever this blood will come in contact with another one or the blood will be transfused to another individual, at that time that individual will suffer. So, it is transfusion of infected blood. Second one, use of contaminated needles, razors like that. That means if one individual is suffering from AIDS and during use of needles or razors, if blood of that individual will be contain or will be contaminated with that of a needle or razor, and that razor or needle if used by a healthy individual, then this virus will pass to that individual. Again, sexual intercourse with one infected partner, if one is suffering from this disease and he come in contact with another healthy person, sexual contact, at the time it will pass from infected mother to the baby through placenta. Then artificial insemination, if in one individual or if the insemination is occurred and that that is this sperm contain the infected or the sperm contain the virus, then it will enter again organ transplantation. That means what I want to say, if one individual is suffering from this type of disease containing this HIV virus and this virus come in contact with the blood of the another one, at that time that virus will be introduced to the healthy individual. This is the transmission. So, once it is transmitted to the body, then how it will work? So, once it is transmitted, then this virus will attack the macrophage cell of the host. The virus enter directly in the macrophage. Inside the macrophage, what will be done? Inside the macrophage, the RNA of the virus replicate to form viral DNA. The very important thing is that we know one DNA replicates or one DNA produces a new DNA by replication. One DNA produces one RNA by transcription. So, the RNA only can be produced by one DNA through one enzyme or by help of an enzyme, that is called as transcriptase enzyme. But in this case, as this virus possesses or as the virus contain one reverse transcriptase. So, what this virus can do? This virus can produce one DNA from the RNA. It is a reverse process. So, once this virus enter into one host cell, inside the host cell, the RNA of the virus will replicate to form one viral DNA. So, this is called a reverse transcriptase, or this is the reverse transcription method, and this is conducted by the reverse transcriptase enzyme which is present within this virus. Then the viral DNA, once the virus produces the viral DNA inside the host cell, then the viral DNA will incorporate to the host cell, and once it will be incorporated to the host cell, then the host cell will produces a number of viral proteins. Understand? So first the RNA will produce the DNA through the reverse transcriptase enzyme. That DNA will be incorporated to the host cell DNA, and inside, once it is incorporated to the host cell DNA, then it will replicate, and this also transcribe a number of proteins, and finally the HIV then enter to the T helper cell, produces a copies of viruses destroying the T cells. So since the number of T cells is reduced, the person loses its immunity. So now you see, for example, this is the host cell, this is the virus. This virus contain, you see, the 2 RNA, and these 2 are the RNA transcriptase enzyme. So once it is introduced, you see this is the viral RNA. This viral RNA by help of reverse transcriptase produces the viral DNA. Then this viral DNA is introduced. Viral DNA become double stranded. Then the viral DNA is introduced into the DNA of the host, and finally this is the host DNA along with the viral DNA. Then this DNA will command, or this DNA will form different type of proteins which are required for the virus. So once the different type of proteins are produced by this viral protein or viral DNA, then a number of virus will be produced. So now in this picture you see again, so up to this you see this is the virus, and this virus again the viral DNA is produced by the reverse transcriptase. So once the reverse transcriptase produces the DNA, this DNA is incorporated with the host DNA. Then this host DNA produces a number of proteins. These are called as viral proteins and viral DNA. So once a number of viral DNAs and viral proteins are produced, then this DNA will again produce the RNA, and that RNA will again incorporate with the protein and from the viral particles. So by this way, this destroy one by one T cell of the body, and a time will come when the number of the T cell will be reduced. At that time this individual will unable to sustain the life, because the B cell cannot produce any type of antibody whenever any type of organism or any type of microbe will introduced into the body. So this body will cannot or are not able to produce the antibody. So this is immunodeficiency syndrome. So this is all about the what? All about the type or all about the transmission of this virus. So now symptom. So when one individual will suffer from this disease, at that time symptoms are fever, then pharyngitis, then nausea, rashes, headache. That means as one individual is unable to exhibit his immunity, so different type of diseases will come to the individual. That means that disease cannot protect the virus or protozoa or bacteria or antigen which enter to the body, because it has no immune system. So different type of disease will be encountered by that individual. So especially fever, pharyngitis, nausea, weight loss, rashes, headache, like that. Again, this disease can be diagnosed by one method, by one enzyme based method, that is called ELISA, or this is called as enzyme linked immunoassay test. So again another method is there, that is called as Western blotting test. By this in one individual again can be tested as the AIDS positive. And up to now we know the virus can be treated, virus cannot be treated through any type of antibiotics. Still some medicines are prescribed which are called as Zidovudine or AZT. So these are the medicines which are given. So prevention. So how this disease will be prevented? So first one, people should be educated. So as we know, one individual when suffering from this disease, at that time there is the possibility of transmission of this disease from one individual to the other when the blood will come in contact. So it will be aware, or we should know about this one. What is the actual exact cause? So people should be educated. What about the AIDS? And especially we are observing one day that the December 1, which is actually observed as AIDS Day. December 1 is observed as AIDS Day only to bring awareness among the people regarding the AIDS. Second one, we should be restricted to the monogamous sexual relationship. So if one individual is suffering and that individual is again using, or that individual again they take the shelter of polygamous method, that means intercourse with different individuals, at that time this will begin spread. So monogamous sexual relationship to be maintained, avoid common blades and barbers shop. So that should be not common, these are different. Along with that, National AIDS Control Organization is there, which is called NACO. So this organization is working, that is called as National AIDS Control Organization, that is called as NACO. So this organization of India is again controlling and even rehabilitating the individuals for recovery of this AIDS. So this AIDS is not one a disease that should not be avoided from the individual. That means we should not avoid the individual which is suffering from this disease, rather we should help the individual how this individual will be isolated, how the individual will be cured from disease. This is actually educated by the National AIDS Control Organization. So this is all about the AIDS. And the last portion of this unit is about the adolescents, drug and alcohol abuse. So this is the last part of this unit. So now we shall see, so what is actually adolescents. We have studied during our previous classes that whenever one individual will sexually mature at a particular age, at that time we can say that this individual is adult, and this period is called adolescence. So adolescence is the period of rapid growth, that means up to after birth, whether it is a male or female. So growth will continue, but a particular time will come when the growth will be very rapid, physical mental change will happen, and finally this individual will transform from childhood to the adulthood. This is called as adolescent, or this is called as inset of puberty. At that time this individual, that means if it is a female, then this female will, that means exhibit the menstruation, and if this is a male, then there will be the coming out of the sperm from the male tract. This is called as puberty. So it is the period, or adolescence is the period of rapid growth and physical and mental development between childhood and adulthood. That means we have one childhood age, that means childhood period, and one adulthood period, and the transition one, that means the age from which the childhood is jumped to the or spring up to the adulthood is called as adolescence period. So it happens in the period between 12 to 18 years. It may happen before 12 or around 12, or it will happen in 13, 14 like that, maximum in 18 years both in male and female. So this period is from puberty, and the period will be counted from the puberty, that means whether it is male or female, when the puberty will start and finally it will achieve the complete sexual maturity. So this is called as adolescence period. So it will take 2 to 3 years, that means if it is 12, then 12, 13, 14 like that, so completely it will be reproductively mature. So what happens when this period come, that means when adolescence period come, what happen in the, or whether it is a male or a female, whether it is a boy or a girl, so curiosity and excitement are common causes. Curiosity and excitement are the common causes which motivate young boys and girls towards drug and alcohol use during this period. So the most important thing, why we are going to discuss this one, because this is a very important period. During this period, due to some hormone secretion, what happen inside the boy or girl, some curiosity to know, to make some experiment, to know everything. So this type of psychological thought comes. So this is called as curiosity and excitement are common causes which motivate the young boys and girls towards the drugs and alcohol use during this period. And at that time, unsupportive family structures may be associated with drug and alcohol abuse among adolescents. Then in this slide, just 1 to 2 slides are there. In this slide we shall see how the adolescents are being suffered by drug and alcohol abuse. So you see what is a drug abuse then. So all of us we know drug is a chemical which is used to cure some disease. That means whenever I am suffering from a particular disease, at that time I have been administered with a particular chemical, that is called as medicine, that is called drug, like that. So the drug refers to a chemical which is usually taken for the treatment of disease advised by the physician. But prolonged use of some drugs even after the cure of the disease can lead to the addiction, and that is called as drug abuse. So use of drug other than the medical use that impairs physical and physiological function during this or drug abuse. That means if a drug is given to me and I am been cured, and after cure if again I continue that drug, and this I become the addicted. So at that time this is called as drug abuse. So different type of drugs are there. So some commonly abused drugs are, which are actually which are actually taken by the adolescents, that means the young girls and boys. They are opioids, then coca, then alkaloids, then barbiturates etc. So these are the drugs. So now we shall see different type of drugs are there. They are sedatives, they are stimulants, they are hallucinogens, they are opiate and narcotics. So sedative is a drug which actually depresses the activity of the central nervous system. Example is benzodiazepines and barbiturates. Stimulants generally induce the excitement by increasing the activity of the central nervous system. Example are caffeine, cocaine, amphetamines used by the athletes can be detected by dope test. So athletes generally use this type of drug. Then hallucinogens, they actually change the feelings and thoughts. So examples are lysergic acid diethylamide, that is LSD. This is one drug. Hemp plant products like charas, bhang, ganja, like that, these are also drugs. Similarly, opiate narcotics suppress pain, tension etc. So these are also drugs. So opium, morphine, brown sugar, these are the drugs. So these type of drugs are generally taken by this youngster or youngs to relieve the pain. So these type of drugs are generally abused. Similarly, the alcohol, which is ethyl alcohol, commonly consumed by the society or the members of the society, they are whiskey, the rum, the brandy, the gin, the wine, beer etc. So these are actually not good for health. So a very little amount is good for health, but if this is habituated, then this is called as alcohol abuse. So dependency on alcohol is called as alcohol abuse. So what is the effect of drug and alcohol abuse? That means during the adolescence period, if one take the shelter of this type of drug and alcohol, then what will happen? The drugs actually suppress the immune system, and the brain are the effects of drug abuse. So immune system is destroyed and brain are being affected by the drug abuse. Similarly, if one alcohol abuse is there, then this causes gastritis, then causes hepatitis, then effect on heart and kidney which are related to the alcohol abuse. So in this slide I just show you what type of drugs are there and what are the effect of the drugs to our body, and alcohol, the alcohol abuse, like that. So this is all about the drug and alcohol abuse. Thank you.